Hi, I'm John St. John. I'm one of the co-founders of Driver Group. We're a company that basically takes the tumor and the normal from late-stage lung cancer patients, and from that data we give patients a clinical report that tells them exactly what kind of drugs they would most benefit from. So today I'm going to talk about what that data is, how we collect it, and what it means. So in this talk today I hope to teach you all about that, and also at the end I'll be showing a little promo slide. We are hiring, so if you are interested in this kind of stuff, please talk to me. So I'll start by talking about what cancer is at the molecular level. I'll then talk about how we turn just tumors, raw tumor samples, into text. Then we'll go into what the data means, how it's actually helped patients, and then I'll go into some next generation analyses, like actually modeling what's happening inside of a cell and what that looks like, followed by opportunities for learning and getting into the field. All right, so cancer is a disease of normal cells in your body that just start going bad, okay? So normal cells in your body know how to act in, they know how to behave. They don't try to grow uncontrollably. They don't try to outcompete their neighbors. They're, well-behaved cells. In tumors, that's not the case. They forget how to behave nicely, and they start dividing like crazy, and they start forming giant masses and spreading all over your body. So basically, it's like a little piece of you that is now trying to take over. Luckily for you, there are a lot of things that have to go wrong before you get a tumor. Okay, so these are the different things, the different kinds of biological processes that actually need to change before you develop cancer. So just as an example, I'll focus on one of them, which is avoiding immune destruction. So the immune system works by, you've got these cells called dendritic cells. They just float around inside your body and wait for little bits of protein to float around. And if it finds one of these little bits of protein, and it is not a bit of protein that it recognizes, remember tumors are filled with mutations that causes different kinds of proteins. If it's not a kind of a protein that it recognizes, it'll let a T cell know that, hey, I found this thing floating around. This is bad, you should go look for it. So now a T cell, if it's behaving the right way, will go and it'll actually hook up with the tumor and it will destroy the tumor cell. So your immune system is actually capable of destroying cancer if it's operating functionally around the tumor. So tumors have to avoid that. Again, that's one example of quite a few different things that all need to go wrong before you actually develop a tumor that's going to kill you. So Frank mentioned DNA. It's huge. Something really crazy to think about. Cells are so small you can't even see them, right? They're like tiny, tiny, tiny things. If you were to take all the DNA in your cell, in just one cell, and stretch it out, it would be 1.5 meters long. And that's like the physical length of the thing. DNA is composed of A's, C's, G's, and T's, represent little chemicals. It's only 10 atoms wide. So it's a very, very small thing. It's kind of remarkable to think that we can even sequence this stuff and even see it. This is pretty new technology. It's maybe 10, 15 years old that we've been able to do this really fast. So it's an exciting time to be involved in biology right now. So what goes on in a tumor? I told you a lot of things need to change. A lot of things need to go wrong before you can get a tumor. Well, you typically accumulate mutations. So it's usually not one mutation, it's usually a large number of mutations. What is a mutation? It's typically a change in your DNA. So this is representing a person's normal cell and a person's tumor cell. And you can see right here there's a little chunk of DNA that has been inserted. It's been added to the person's tumor. That could potentially cause the tumor to start misbehaving in one of the ways that leads to cancer. So I don't know how many of you have heard about the central dogma of biology. It's this whole idea that you've got DNA, your genome, within your genome, little pieces of your genome encode RNA, right? So this is zooming in on a gene. This is zooming in on a piece of DNA, which encodes RNA, which encodes a protein. A protein is a little glob that floats around and actually does stuff inside of your body, okay? So really it's the proteins, that you have to be interested in. But as a limitation of current technology, we can't look at proteins directly very easily in large numbers. So we typically focus on DNA and RNA, which are easy to read. So what can go wrong? What does a mutation do? Well, as Frank mentioned, the typical things that can happen, you can have either an underrepresentation of a gene, so not enough of a certain protein could be present, and that could be bad in a cancer cell. You could have too much of a gene. So for example, say a gene that's involved in developing embryo, right? So you could imagine that that would be bad if all of a sudden you're expressing developing embryo genes as an adult, right? So that can happen in tumors. You can also just have qualitatively different, or just different proteins, different shapes of proteins that now behave differently, and maybe they don't interact with other proteins properly. So that's what's represented down there. So how do we turn this into text? How do we turn this into something that we can actually analyze? So this is DNA sequencing. Typically what we'll do, and this is starting to change, people are starting to look at the whole genome, everything involving genes and also everything between the genes. But typically what the medical field does today, including our company, is we just extract the parts of the genome that turn into proteins at some point. So this is what we call the exome. So instead of 3.2 billion characters, we're down to 55 million that we have to look at. So still quite a bit of data. And then in addition to looking at the DNA from the exome, we also look at what it turns into, the RNA, as a proxy for the protein. So it gives us some idea of how much protein is being expressed in a cell, how much protein is present. So these are sort of the two different kinds of data that we typically look at. Both of these kinds of data, we can, biological data, we can feed them into sequencers and read them the same way. At the end, they create the same kinds of files. So how much data is this? Why do we need Spark, right? So this is War and Peace. It's a really huge book. I haven't read it. I just know it's huge. If you were to replace every single letter in this book with an A, C, G, or T, 1,400 pages long, 3 million letters, you have to sequence a lot of these books. Okay, so we're looking at about 6,000 copies of War and Peace because of all of the redundancy that we need in this data. And unfortunately, one limitation of modern technology is that you can't read the DNA end to end, right? You can't just read it, position 1 to position 1,000. First, you throw it in a paper shredder. So imagine taking like several bookshelves filled with books, throw it in a paper shredder, and then what does this represent? Well, now you got to find some mutations, right? What are mutations? They're little typos. You have to find tiny typos, maybe 100 of them in this huge pile of data, right? You have to find slight shifts in DNA abundance. I'll talk about that in a minute and what that means. Slight differences in RNA abundance. I'll talk about that as well. But really, it's like there's so much noise here and there's so much data here. You have to have some pretty powerful methods and a bit of knowledge about how to really process this the right way. So now let me introduce you to the data format that you start with. It's called FASTQ. There's absolutely nothing attached to it, so none of the nice stuff in Frank's schema. You don't know where it belongs, you don't know where it comes from. All you know is a unique identifier that says that this sequence has some kind of name and it's different from other sequences. The DNA sequence itself, typically about 100 nucleotides long with modern technology, and then confidence scores for each letter. That's typically what you get out of modern sequencers. So now I mentioned the first thing that we're looking for, on the order of about 100, maybe to 1,000 mutations in a typical tumor. I didn't mention before, but we're actually looking specifically at lung cancer. Someone asked about how much, aren't tumors really messed up? Well, it varies quite a bit. All right, so lung cancer is actually one of the more messed up kinds of tumors. Lung tumors, you typically have mutations all over the place. It's one of the most mutated tumors. So I guess we're lucky in a sense that we're analyzing lung cancer because we see a lot of these, so we have a lot to work with. So what we typically do is a comparison between a patient's normal and their tumor. So that gives us this diff that Frank mentioned a few times, right? So what I'm showing you here is an actual mutation in a gene, EGFR, that's related to lung cancer. And if you have this mutation, that means that you're eligible for a certain lung cancer therapy. And it will extend those patients' lives about an extra 6 months over standard chemo and radiation. So it's a big deal if you can find this. But you're looking through a lot of data to get there. You have to have pretty sensitive methods that are also very specific. You know, false positives are huge when you're dealing with like 50 gigabytes of text data filled with errors. So I also mentioned copy number changes. Chunks of your chromosome can get duplicated. So remember earlier we were talking about genes and expression levels of genes. Well, one way to get a higher expression level of a gene is just put it there twice, okay? So this happens all the time in tumors. Literally a chunk of your chromosome, which is, you know, part of your DNA, will just copy itself and it'll just be there twice. So how do you see that, right? I mean, it's not actually like changing any letters anywhere, right? But what you can do is you can just count how much data you have in a given window in your tumor sample and in your normal sample. And you can do a ratio, and you can slide that window along the genome, and you can look for significant shifts in the ratio that are consistent, right? So you can see there's tons of noise everywhere, but you do see these very large, very significant shifts. So that's what copy number change looks like. And then I mentioned RNA. This one's tricky. This is maybe one area that is, most clinical labs don't even want to touch it. It's really tough to deal with. It's very noisy data in the expression level. It's hard to interpret. We are attempting to go there right now. Our lab is. So there's a lot of work to be done, but I'm hopeful. There are certain kinds of things that we can do right now with it, for example. So we can classify what kind of tumor you have just based on the RNA data, which is pretty neat, just to see that kind of a biological result just fall out of this data. But you know, what is this data, right? So just like in copy number change, what you can do is you can look at the RNA and you can count how many reads in the RNA match up with the tumor versus the normal. So it's another ratio kind of problem, right? And if you plotted out every single gene and what it looked like and just highlighted the outliers, you could look for certain genes where there's way more of it in the tumor than the normal, okay? If it was exactly identical, it would be on this line right here. But what we see instead are, you know, some genes that are high magnitude outliers that are really shifted, where you've got a way higher abundance in the cancer sample than you do in the normal sample. So those kinds of things are interesting, as well as these, you know, really low abundance RNAs. So how's this actually helped people so far? So here's a real case study from a few years ago, a drug called Erlotinib. In initial clinical trials, which were not targeted in a patient-specific way, they noticed that the drug worked really well in some random 10% of the people. They didn't know why. It didn't seem to do anything to the other 90%. So, you know, just looking at this from a maybe 20-year-old like oncology company point of view, this would be a failure. This would be a failed trial. What they noticed was that also about that same proportion of patients have a mutation in the gene that that drug is theorized to target. Okay, so two separate pieces of data. Now, if you put them together and you condition one on the other, you'll actually see that all of the response to this drug is in that class of patients that have the mutation. So that led to the current standard of care, which is that every single lung cancer patient that goes to a hospital gets tested for that specific gene to see if they have that mutation, and if they do, they get this drug, and it helps them out quite a bit. So here's something that just happened. immune therapy. This is like the new hot field in cancer therapy right now. I mentioned the immune system before, how that's one of the many things that has to go wrong in a patient's tumor before it's actually a successful tumor. And the results are really interesting, and they look a lot like the results for Erlotinib did a few slides ago. It works really well in about 17% of the patients. In fact, I believe in a good percent of that 17%, the patients just completely stopped, and for the duration of the trial, they haven't continued to develop more tumors, which is pretty remarkable. So this is a pretty exciting one, but it's just 17%. Why is that? Why is it just that 17%? Unfortunately, we don't know. Current stratification methods have been unsuccessful. So that brings me to the next topic, which is pathways. So how do you actually model a cell? Most cellular things aren't simple single gene stories. You've got these things called pathways. So here's a really simple example of a pathway. On the surface of a cell, you've got some gene called RTK. When there's a lot of that gene, it can signal this gene called PIK3CA. When there's a lot of that gene, it can signal this one called AKT. When there's a lot of that, it can signal cell survival and proliferation. So what happens in tumors? Well, in a lot of tumors, you actually see a mutation in this gene called PIK3CA. It's what I call a gain-of-function mutation. A lot of people call it that, not just me. Gain-of-function mutation, it activates this interaction right here and any other interaction that I'm not showing in this graph. And as you could imagine, that has downstream effects. Okay, so now it's not just one gene interacting in isolation. It's now a whole cluster of genes. It's a set of genes acting in a network, interacting with each other, talking to each other. This is more of what tumors actually are, right? This is what's happening inside of your cells. So how do you identify these pathways? There are a couple methods. There are a bunch of different ways you could go. You could just look at the RNA levels. You could try to do that without any idea of what the pathways are. I could show you what that looks like in a bit. You could also try to take advantage computationally of known pathway structures. That's actually a little tricky because often pathway structures aren't complete. There's a lot of missing data. And also in certain cells, pathways are actually different than they are in other cells. Like different genes will talk to different genes. It's kind of confusing. So there are drawbacks to that approach, even though it sounds a lot better. There are also things you can do with DNA just by itself. And there are methods out there now that can integrate both DNA and RNA at the same time and try to model the cell more completely. But again, for the same reasons, it's not clear which one of these methods is going to be the most productive, and chances are it's going to differ in different cases. So first off, I want to talk about RNA without pathways. This is surprisingly useful kinds of analysis right here. This is how we are currently able to predict what kind of cancer someone has when they come in. About 4% of all cancer patients, the doctors aren't able to figure out where the cancer originated, which can actually change what kind of care the patient gets. So it's pretty useful information. Different cancers use different genes, right? So different cancers will use a different set of genes to be active. And what you can do is through feature selection, gene selection. This is showing differential expression where red things are way higher in the tumor than normal, 3 standard deviations for the most red, or way lower in the tumor than normal, -3 standard deviations for the most blue. And what you can see is that different tumor types very consistently tend to use different sets of genes. So if you're really careful with your machine learning and you avoid overfitting because there are 20,000 genes and maybe only about 200 patients or so, what is that? N greater than P or P greater than N? I forgot which direction it is, but it's the bad one. So yeah, but it is very useful if you're careful. You can also take advantage of the pathway diagram itself. And when you do this, you're typically looking for consistency with the pathway structure. So for example, just showing a subset of this gene here, AKT1, since there's a lot of it, this right here is not an arrow, this is a blocking a blocking relationship. So if there's a lot of this, you'd expect there to be not very much of this. So you can do these kinds of consistency checks with different sets of genes and their known relationships or their assumed relationships, and you can score based on that, and then choose which pathways are being perturbed the most based on these kinds of consistency relationships. So I talked about DNA-only methods earlier. The problem here is that pathways have a lot of redundancy. So this is the pathway I showed you earlier. Now imagine you had, I showed you this specific interaction here where you had a gain-of-function mutation in PIK3CA. That has the downstream effect of cell survival and proliferation, and it works like that. You can actually have a loss of function in this gene with the exact same downstream effects. It works just as well for the tumor, so why should the tumor really care? Similarly, you could also knock out that gene. Same downstream effects. Why should the tumor care? A lot of times the tumor doesn't care, and it just gets one of those randomly. So if you're doing a statistical test and you're only looking at one gene at a time, you have much less power because all of your signal is just randomly split up between all of the redundant events, right? So if you take the knowledge of the pathway diagram and you take the individual events that occur and you spread the influence of those events through the pathway diagram, and then you can use that as your input to standard clustering methods or other kinds of machine learning methods. So there's a lot you can do with pathways just based on DNA by itself. So I mentioned integrated methods. So this took me a little while to understand, but this is pretty cool stuff. So what this is showing, what I want to show you right now, is that there is signal there. There's signal between DNA and RNA. When something happens in DNA and you have a known relationship between one gene and another gene, gene B, the second gene in that known relationship, the downstream gene, if you expect that gene to be, say, turned on by gene A, you can look at gene A being more abundant in the DNA, and you can observe gene B being more abundant in the RNA, right? So if you think about that for a second, that validates the idea that A, the copy number is actually having an effect on the expression of gene A, and it validates the interaction between gene A and gene B with a correlation between the two expression levels. And similarly in blue here, if you just look at the expression of A and the expression of B, you also have higher than expected correlation out of the two. So these pathways do store some real information, and you can actually see it. It's tough though. I mean, the method that does this, there's a whole company built up around it. It's not a trivial analysis. You have to know quite a bit about, about the different kinds of interactions and how they work together, and you have to model the thing properly. And I think one of the companies I know of that does this, they use a Bayesian belief network to do it. It's doable, but tough. We should put it in Spark. All right, so what do you do now? I don't know. Maybe some of you are interested in pursuing this more, maybe not. One site that I really like is called rosalind.info. They have a lot of different just little toy problems along with explanations for what those toy problems are. You can go through that, do some code, just kind of get a feeling for what the data looks like and what it means. I think it's pretty useful. I mean, it might be a bigger jump between that and actual data analysis than some things, but they do actually have some really, some more complex algorithms in there, so it's not totally trivial stuff. Coursera, we mentioned. I didn't actually know that that one guy was teaching a class. That's really cool. I'm probably going to take it. These classes are useful. This is our continuing education, so all of us in genomics pretty much do this. There's one called this bioinformaticsalgorithms.com. There's a book. There's some Coursera classes. That's the group at UC San Diego. Pavel Pesner, I believe, is the guy's name. He's like really, really good in the field. There's this group at Harvard, that has a whole bunch of things, including a class on variant discovery and genotyping, which I had a hard time finding classes on. So, so that's a cool example. Full disclosure, this isn't my background. This isn't how I learned. I was like midway through a PhD in this field, and then I dropped out to join this company. But anyways, I have done these kind of classes to learn more stuff about things. And obviously, Frank's website, bdgenomics.org, avocado, pick-me-up issues. I've done a little bit there. It's totally doable, and Frank is very helpful, so definitely do that as well. And of course, you can come work for Driver Group with me. We're hiring DevOps, sysadmins, software engineers, data scientists, data warehousing people, actually robotics automation specialists. If you are or know anyone who knows that kind of stuff, that would be really cool too. So yeah, thank you. Questions? Any questions? Yes?
Hi. So if you go over the pathways, it looked like that was like graph representation.
Uh-huh.
But it may just be graphical.
No, they're graphs.
GraphX.
Yeah.
So are you using GraphX?
No.
This is the question.
So the question is, pathways appear to be graphs rather than just graphical. That is correct. Pathways are graphs. And are we using GraphX? The answer is no. These are methods that we're just now starting to break into, and I believe no one else has done it yet in GraphX. So there you go. You want to do something? Why don't you take some like cool method and put it in GraphX? We'll use it. Yes. Yes. So the question is, what are we doing for robotics? Yeah, actually, it's prepping the DNA and running it through the entire, like, what we would call the wet lab process. So basically, like, all of the, physical handling of the sample up until the sequencing stage. Yeah. It's actually really important. It's not just, like, for the coolness and the scale. It's also really important for consistency. Robots are more consistent than humans.
Really?
Yeah, who'd have thunk? All right, anyone else? And yes?
Another thing, so you were talking about you have a pretty large swath of data you're working with. Are you guys dealing with like primary sequence files for every one of the like different cancer cells that you get, or are you guys like doing diffs on that?
So we do both. I mean, okay, the question was, are we dealing with primary sequence files or diffs? The answer is that, we have, our pipeline starts with the output of the sequencer, which is just the primary data. So, that's on the order of 50 gigabytes for each person in an image-ish format called BCL, and then that gets converted to FASTQ, which is the format I showed earlier. And then we do alignment and processing and all the stuff that Frank was talking about. And yeah, we go through the whole A to Z on that. Yes?
What's it like working with Driver Group? Tell us about a typical day.
What's it like working with Driver Group? Tell us a typical day. Okay. So you get into the office, you look at your super long list of things. We don't drink beer in the morning. We look at a super long list of like things that we still have to do because like we're doing so much stuff. There's so many methods out there that just they're not implemented yet. We're dealing right now with like how to take our lab, what we've built here in San Francisco, and expand it out to China. It's a lot of stuff, but we've got a really fun office. Everyone's really smart. We like working together. We like hanging out. So there's a lot of that, as well as we've actually had cancer patients come by. So it's a very interesting mix of like, serious, oh my God, we're actually helping save lives, with we're having a really good time and doing data analysis and building these giant tools. So I like it. Yes.
You mentioned you're just using exomes. Have you tried, getting your exome whole genome sequencing data PCR free? Is there any benefit to that?
So the question is, have we tried whole genome PCR free is another word you mentioned. Is there any benefit to that? Personally, right now, I'd rather go in the other direction for some technical reasons. I'd rather get even more focused on a handful of genes that we know are more important, just because you never get a pure sample, okay? So if you're doing a research project, for example, your sample's probably 100% tumor cells, right? So every single read that you sequence is coming from that tumor. Now, in the real world, what you get is some crappy biopsy that's not taken very well, and most of it's surrounding normal tissue. Not a lot of it's tumor. It's highly degraded. You got to go deep on that to really see stuff. You've got to resample it a lot. Sometimes, we've had samples come in that are maybe 5% of the sample is tumor, and the rest of it's just surrounding normal tissue. So for that, you really have to sequence deep. So if we were to sequence that deep on whole genome, I'm thinking maybe 100x or so. Now we're talking not 50 gigabytes per person, it's maybe a few hundred gigabytes per person. So maybe we should hire you to help us do that. And also someone really rich to help us store that. Yes?
Back to the question about your day. That just hearing you talk about this, I begin to just muse about it. So there's research papers to read. There's some really knowledgeable PhD level genomic scientists and computer wizards and everything. So what are the conversations like? How do they happen? Are they in front of whiteboards? Are they meeting room discussions? Are they in the hallway? How do those big lists how do things get on a person's big list?
Yeah. Well, we do a lot of the same stuff that a lot of tech companies do with Trello and Slack and all that kind of stuff. We also have whiteboards, and we do a lot of just standing in front of the whiteboard and drawing stuff out and, thinking about algorithms and how to normalize between different tumor purities, between, tumor 1 and tumor 2, and after you've done that normalization, how to correct the data for that so that you can see if the mutation's actually increasing or decreasing in abundancy and not just changing in purity between the two samples. There's a lot of fun stuff that we think about and whiteboard and yeah. Yes?
So I'm really glad you mentioned the investment tumor biopsy.
Mm-hmm.
We were talking about this entire medical stack. There's lots of places for improvement.
Yeah.
Are you guys doing single cell isolation pre-sequencing? Is your prep pulling out individual cells? And say, for the example of the copy number variants, it seems to me like that would be a really good way to solve that in hardware, I guess you could say.
Yeah, it's a great question. So the question was about whether or not we're doing what's called single cell sequencing from patient data. Because that seems like that would solve a lot of our problems sort of at the hardware level, be pre-analytic. How much do you know about how hospitals work in the US and how difficult it is to convince the pathology department to actually give you a tumor sample? It's tough. For next-gen sequencing data like we're doing, we're lucky if we get the dregs from a sample. And that is just deep, entrenched bureaucracy, and it is really hard. So earlier, I mentioned that we are opening up a lab in China. Yep. China does not have that bureaucracy. They are happy to do the most up-to-date, fanciest, newest kinds of algorithmic analysis. We're actually working with a hospital over there to put a genome sequencer right next to the OR. So it's pretty amazing stuff. And we're going to be the first people seeing that kind of data from actual patients. So yeah, come work for us. Yes?
What's next after
That's a good question. So the question was, what's next after lung cancer? Are we looking at other kinds of cancer? It depends on the time frame you're talking about. So if you're talking about someday, yes, we want to do every kind of cancer. The problem is, if you spread your focus too thin, you're not going to collect enough data in any one tumor type fast enough to make a clinically actionable decision about what drugs you should go after. So our company's business model is actually to figure out where there are unmet needs in patient populations with particular drugs. So what we want to do is figure out, for example, that a certain percentage of lung cancer patients have certain kinds of mutations, and we think they would do really well. These particular patients, we think they would do really well if they received this specific kind of drug. So what we want to do is kind of bring all that together. So we're actually going to be running some early stage clinical trials ourselves, and hopefully figuring out drugs that work and then sell those drugs back to the pharma companies. So that's the goal.
Any other questions?